Hello everyone, I'm Tolo M. Cody from Tumor Metabolism and Therapeutic Oncology Research Laboratory. My research project is titled Exploring a Functional Relationship Between CRBN and SHMT2. To study the biological significance of CRBN and SHMT2 interaction is my research aim. Cereblon, commonly referred to as CRBN, is a E3-ubiquitin ligase protein that interacts with the DNA damage protein 1 colon 4 and the regulator of colon 1 to form the functional E3-ubiquitin ligase complex and function as the substrate receptor of this complex. Additionally, the well-known immunomodulatory drug IMIDs implement its anti-tumor activity through the cereblon. CRBN being an E3-ubiquitin ligase protein selects specific substrate for degradation, but in the presence of the IMIDs, CRBN recruits new substrates and degrades it through the ubiquitin proteasome system. In addition to its ubiquitination function, CRBN also functions as a molecular chaperone. A clear function or a clear example of its molecular chaperone function is shown in this paper, precisely in this diagram. So, in here, CRBN forms a complex with HISHAP protein and AHA1. By CRBN counteracting AHA1, it leads to the stability of two transmembrane proteins, CD98AC and LAT1. But as seen here, in the presence of IMIDs, this interaction is disabled and there is no stability. Now over to serine hydroxymethyltransferase known as SHMT2. So in humans, we have two isoforms of the SHMT2 gene, of the SHMT gene. SHMT1 is localized in the cytoplasm, while SHMT2 is localized in the mitochondria. SHMT2 is a major enzyme in the 1-carbon metabolism. What does this enzyme do? It catalyzes the conversion of serine and THS, which is tetrahydrofolate, into glycine and 5,10-methyl tetrahydrofolate. This action gradually leads to the transfer of 1 carbon to the purine synthesis and to other compartments of the cell. Recently, it has been reported that SHMT2 is abnormally expressed in cancer cell lines. So in this paper, to assess or analyze SHMT2 expression with that of human cancers, they use the public RNA-seq data from TCGA database and check SHMT2 levels in comparison to normal across series of cancer types. So as seen here, compared to normal, 85% of these cancer types show high expression of SHMT2, which indicates that SHMT2 could serve as a biomarker in cancer. Also, in order to have more background information on my research, as I am trying to study the functional relationship between CRBN and SHMT2 interaction, I found this paper in which CRBN interacts with SHMT2. And according to these researchers, CRBM associates with about 176 proteins. Of this association or interaction, 10 of these proteins strongly interact with CRBM, one of which is SHMT2. So to come out with this conclusion, they performed the experiment using two different assays, immunoprecipitation and pull-down assay. So this assay shows that SHMT2 strongly interacted with 10 proteins, one of which is SHMT2. Also in our lab, we perform ubiquitination signaling profile of SHMT2, and the result shows that SHMT2 has a strong ubiquitin signal in response to thalidomide. So with this, I check SHMT2 expression levels in various cancer cell lines in which CRBM is knocked out. So as seen here, I check in 3 different cancer cell lines and normal like cell line inclusive. At the mRNA level, there is no change. SHMT2 expression remains unchanged. But at the protein level, in CACI1, a kidney cell line, SHMT2 expression is elevated. Next, I clone the SHMT2 gene into flag plasmids and the CRBM into HA plasmids in order to check that interaction by means of co-immunoprecipitation. So in this result, I did a co-immunoprecipitation of HA, CRBM, and SHMT2. As seen here, clearly CRBM interacted with SHMT2. Also, to confirm this result, I use AMPK, which is well known to interact with CRBM, and AMPK also confirmed this binding. Well, in the bit to confirm my result, I also did a bidirectional co-IP. This time, I used flag SHMT2 to pull down HACRBM, and as seen here, it was successfully pulled down, which shows that CRBM interacts with SHMT2. So to conclude, in CRBM knockout kidney cancer cell line, SHMT2 transcriptional level remains unchanged, while the protein level is elevated about 1.8 folds. Co-immunoprecipitation analysis reveals CRBM interacts with SHMT2. Currently, I'm working on a co-immunoprecipitation assay of CRBM and SHMT2 interaction in the presence of thalidomide. And then I will move on to check the domains involved in the interaction, perform ubiquitination assay, and also check the pathway or mechanism involved in this interaction. Thank you so much for listening, and a very big thank to my professor, Professor Steve Chill, and all my lab mates for the support thus far. Thank you all.